Welcome, everyone. It's a delight to see you all here on this very cold and rainy evening. And I am very happy to be introducing our speaker for the evening, who is Professor Andrew Leitch from Queen Mary University of London. Andrew is a professor of plant genomics, and he has been there for an amazing 30 years almost. Just shy. You didn't know that, did you? Yeah. So he is in the School of Biological and Behavioral Sciences at Queen Mary, where he specializes in plant genomics, genetics, and cytogenetics. And his questions are really broad across ecology and evolution, ranging from hybridization between species to the origins and the consequences of plant genome size, which is, of course, what he's going to be talking about today. Before he was at Queen Mary, he was a postdoc at the University of Cambridge and did his PhD at the University of Bristol. So I'm very excited to hear about Andrew's talk, and welcome up here.
Thank you for that introduction, and thank you also for this invitation in this wonderful room. It's such a pleasure to be in this place. And I've just been told it's the anniversary of the publication of The Origin of the Species. So what an honored day it is to be here. I was told that Linnean society, perhaps I shouldn't spread false rumors, that apparently nothing significant happened on that day. And I suspect nothing significant is going to happen on this day either. We shall see. Anyway, the talk I'm going to give you today is, as the title shows, Genome Downsizing and Polyploidy Mechanisms, Rates, and Selection Pressures. It's a teamwork, and I'd really like to thank the people here, particularly Ilya at the Royal Botanic Gardens, Kew, and our team under my name, under Queen Mary, who are fantastic young scientists, and you'll be hearing more from them later on. In the center of this is a diagram from my US colleague, Jonathan Wendell, and it really summarizes beautifully what this talk is about. It's about plant genomes. They increase in the number of chromosomes. That's called polyploidy. Yeah. No, they're still there. There, oh, still there. It really doesn't want to go away. Yeah, perhaps it has gone away now. Okay, I think we're in business again. Thank you. So this central diagram here, it shows how plants undergo polyploidy. And this happens iteratively in the evolution of the angiosperms. But after polyploidy, the genomes whittle down to a diploid-like form. And so you come back millions of years later and look at those genomes and you think, aha, I've got a diploid here. Whereas in fact, underneath it, there's rounds of polyploidy. So really that diagram is going to sum up the essence of what this talk is about. Okay, so the talk is going to be divided up into these sections. First of all, a little bit about genome sizes and so on, and then how that can lead to ecological filtering based on genomes. And then why genome downsizing happens, why we think it's happening, and where selection might act on it. And then we ask the question, actually, is selection actually possible? And then we summarize it. So there's our journey. So first of all, the introduction. Well, some years ago, Ilya was involved, well, she still is involved in assembling what's called the Plant C-Values Database, which is a collection of the genome sizes of plants globally. And she plotted the distribution of genome sizes. And here on the left axis, you've got the number of species, and on the y-axis, the genome sizes. And that distribution really intrigued me. And in fact, when I first saw that and really started thinking about it, it really did change the course of what happened in my laboratory and what we worked on. And what's so interesting to me, at least, about that distribution is that most plant genomes are very small. and they're really banging up against a genome size close to naught. In fact, the modal genome size is about 0.5 gigabases of DNA. But right at the other end there, you've got this long tail stretching out to over 140 gigabases. There's this thing here called Paris japonica with absolutely humongous genomes. And yet these plants, they're all the same, really. They've all got flowers and leaves and so on. So, what exactly is going on in that? And why is it such a skewed distribution? Now, I was told by the mathematicians that this apparently skewed distribution could be occurring because you have a normal distribution bounded by zeros. You can't be less than zero. But in fact, that really doesn't explain that distribution because, as we see later on, other organisms have different distributions, other groups. Well, why have we got this big range? Well, it really comes from 2, 3 perhaps predominant processes. The first of it is that the majority of these plants, well, all these plants have got broadly the same genes. Of course, there's lineage-specific genes and there are gene duplications in different number of gene families, but to a first approximation, things are very similar. And where they're really differing is in the amount of DNA between the genes, and it can be hugely different. And that amount of DNA is comprised of repeats, and repeats that have degraded so much that they're now single copy sequences and no longer repeats. And so the first mechanism we have is repeat amplification, which is what's shown by that yellow bar. But the processes which amplify DNA has at heart recombination processes. They can also delete DNA. And so the balance between enlarging and deletion, amplification and deletion, will affect genome size, depending on which of those processes is outweighing the other. And the other major mechanism underneath it is polyploidy. That's whole genome duplication. Many species have got polyploid cytotype individuals in their population. Others, the polyploidy events actually generate species. And this phenomenon, of course, is very important in the divergence of angiosperms. But of course, it has the effects of amplifying genomes. So I said to you earlier that not all distributions are skewed and small genomes. So the sister groups, the angiosperms, the other seed plant lineage of the gymnosperms, they have a much more normal distribution. The genome sizes are larger, they're much larger, in fact, and there's no hint of this sort of massive long tail or skew in that data set. And of course, they begin with a seed plant ancestor. So something different is going on down that lineage running to what we call angiosperms. What we've learned over the years is that the genome size, the amount of DNA they have effects on the phenotype of the organism that transcends just the effects of the genes they have. That physical bulk of DNA actually has its own properties. And you can see it very nicely here in these guard cells. So what we have here is stomata and stomatal pores. Let's focus on this one in the middle here. There are 2 guard cells, a pore in the middle, and you've got the epidermal cells of the leaf around it. Now, these guard cells each contain a nucleus. And when the nucleus is small, the guard cells are small. But as the nucleus gets bigger, the minimum, because of packing constraints, the cell gets bigger with it. And so at this top end here, you've got the absolutely massive guard cells carrying a massive pore, and the epidermal cells around it are also bigger. So as the guard cells are increasing in size, the pore size is increasing and the density of stomata are dropping off. This is all going to affect the properties of that tissues in this organism. You can see this laid out in some very nice sort of correlations between guard cell length and genome size, and there's a linear correlation between genome size and stomatal density, and it's an inverse correlation, all the stuff you can see in here. And it's not only guard cells this applies to, you can see it in other tissues too, like here in the vein density. Now, this is going to affect the water balance of the cell. Now, we know that a little cell like that can do all sorts of tricks to make itself bigger, like bigger vacuoles and so on. But these ones with the big nuclei cannot get smaller because there's a minimum cell size. You know, there's that packing of that DNA. And so therein lies a problem for the plants with a big genome. Okay, so what about that? We can show those in sort of morphological features. We can see that these effects do occur across the physiological and the deeper biochemistry of the plants. So what we did here is we just went to the TRI database, which has got measurements all done independently by hundreds or thousands of groups around the world, where they're measuring rates of carboxylation. And we just took those measurements and plotted against genome size, and we get that correlation. There is a negative effect. It's noisy and weak, but it is significant. The same with stomatal conductance, noisy and weak, but significant. So we do have trends in here, but it's quite obviously, it's complicated. The genome size is having an impact on these fundamental processes. Well, now let's just dive in a little bit into the ecology and how genome sizes and polyploidy can affect ecology. Well, the first experiment of this nature was done by Schmader et al. In 2013 on what's known as the Regen grassland experiment, which is a fertilizer experiment. And they added fertilizers to these plots since 1941, and they wanted to ask what effect does it have on the species there and the genome size those species had. And then in 2016, we did the same thing at Park Grass experiment and came to broadly the same results. And I'm going to explain the Park Grass results here because, of course, I know that one the best. As you walk onto that experiment, the first thing you notice is that 150, 60 years of fertilizer has made a big difference to the community of the plants that grow on those plots. And you can see it in this lovely early summer photo. Here, where there's no fertilizer has been added in 160 odd years, it's a herb-rich community. Over on the left here, we've added nitrogen. You can see there's a lot more grass, there's a lot less herbaceous plants in general, and it's a lot greener. At the back here, something white has come in. Over the back here on this plot here is obviously a loss of ranunculus or something like that. You can see that adding different fertilizers and different ratios and so on affects community composition. It's long known, of course. And here you can see the effects of fertilizer. This is in fact why we add fertilizer. If you plot total biomass on these plots, you can see there's not much biomass. You add nitrogen, it hardly makes any difference. You add phosphate a little bit more, showing that these plots are probably a bit phosphate deficient. But you add nitrogen and phosphate and bango, fertilizer affects loads of biomass production. So we were then interested in asking the question, what about the genome sizes of the plants on here? When we first walked out onto those plots, we walked out on the low nutrient plots, we were rather horrified to find Fritillaria meleagris, which has the biggest genome of all the species in the British flora. So we rather thought, oh dear, well, let's pack up our bags and go home. But of course, we thought, well, we better take our measurements and see what happens. And as we were doing that, we realized that Fritillaria meleagris was there, but its contribution to the biomass on the plot was vanishingly little. So it was, you know, it was just not important for that. And so we produced this measurement of biomass weighted genome size, where we weighted the biomass, the genome size by the contribution it made to the biomass on the plot. And with that measure, you can see that you add nitrogen and phosphorus and the biomass weighted genome size shoots up. This means more species with a bigger genome are contributing to the biomass on those plots. We then drilled into this a little bit more, and we split the species into two groups, those less than 5 picograms in their genome and those more than it. And you can see that when you add nitrogen and phosphate, it's the plants with larger than 5 picograms genome sizes that are doing well on these plots. And if you split it by diploid or polyploid, that's chromosomal ploidy level, you know, how many copies of the genome they are, you can tell under a microscope, you can see that it's polyploid plants that are doing well. And when you combine them both, are they polyploids with big or little genome? It's the big genome polyploids that are doing well when we add fertilizer. And so that was quite an interesting finding. And to this day, we don't really understand fully why it's polyploid plants with big genomes are doing so well. We've sort of got a pretty good explanation for big genomes, but perhaps less for the polyploid bit of it. And I'm going to come to the big genome bit soon. More recently, we've done the same thing with a lovely group of scientists in China working on Inner Mongolia field trials on a humongous scale experiment in Inner Mongolia, as you can see here. The design of the experiment here is a little bit more complicated, and I'll explain that in the next picture. They're adding nitrogen and phosphorus, or a control with neither of those two, in combination with varying numbers of other nutrients to try to get some handle on whether other nutrients beyond nitrogen and phosphate, what effect that has on the ecology of these ecosystems. And so what I'm going to show you is the effects of nitrogen and phosphorus with varying numbers of other nutrients. Okay, so here's the first thing. On the left axis here, we've got the biomass production. ANPP is simply biomass production. And we're dividing up into three groups then. The gray groups are no nitrogen, no phosphates. The pale green is adding just nitrogen, and the dark green adding nitrogen and phosphates. And then 1, 2, 3, 4, 5, 6, addition beyond nitrogen and phosphorus. And you can see, essentially, the effects are coming in from nitrogen and phosphorus. So you add nitrogen, the biomass goes up. You add nitrogen and phosphorus, it goes up a bit more. When you look at, split this now into large and small genomes, as we did before, you can see that the large genomes, the biomass goes up. With nitrogen, it goes up a bit more with nitrogen and phosphate, but nothing's going on with the small genome size plants. Now, in both the previous experiment and this, of course, we did it using genome size as a continuous variable and phylogenetic correction, and the results survive both of those. Okay, when we now look at it, ask a question, you know, here they are, they're growing nice and high here. The light coming through is slowly getting filtered out to the bottom. So we can ask how much light is being filtered as it moves through the grass canopy. And you can see it's the flip, of course, over here. When you add no nitrogen and phosphorus, there's less canopy, more light gets through. When you add nitrogen and phosphate, the canopy is denser, most of the light is cut out. And so when you're adding fertilizer, the plants get more vigorous, there's more biomass, the more light's cut out. And so that's what this result here shows. When you start looking at number of species there are, on the left axis now, we've got the number of species. We have a result that's the inverse of the biomass production. So what it's showing is that when you have no nitrogen phosphorus, you have more species. You add nitrogen, you get less species. You add nitrogen and phosphate, even less species again. We've known this for a long time. You add fertilizers, species drop out. But when you split it up into large and small genome size species, what's dropping out are the small genome size species. You can see here, this is the one where they're more numerous without any fertilizer and less numerous when you do add fertilizer. So from these data, you can start building up a model of what's going on in the system. And this is the best fit model we have for this data set. And there's Yang Peng, who was in charge of the work out there. So you add nitrogen and phosphorus, and the biomass of the large and the small genomes go up. But the large genomes actually inhibit the growth of the small genomes. They like the phosphate, the small genome plants, but then they're not doing well in competition with the small genome plants, which in the presence of the large genome plants, which is having a negative effect upon them. As the large genome plants put on biomass, there's less radiation reaching the surface. The canopy is getting bigger, and so it's cutting out the light. The cutting out of that light has a negative effect on the richness of the small genome size plants, because there's a positive correlation between the amount of light reaching the surface and the richness of the plants. So what you have here then is that drops off, the light drops off there, it has an effect of dropping it off in the richness of small genome size plants. So this looks like the dynamic going on in this system. We extended this because we now got this happening in Inner Mongolia and 2 European sites. And there's a lovely collection of field experiments, nutrient network experiments of field trials adding nitrogen and phosphate across the world with sites in North America and across Europe. And we teamed up with Erica Hirsch-Green and worked out what's going on globally across these nutrient network plots. And our super students, Joe Morton, came up with these data. What you have here is the change in weighted genome size, much the same as the type of weighting we had previously, when you add nitrogen, phosphorus, and nitrogen and phosphorus. So across all these sites, this is the effects that's going on. It's the same thing coming out, the same again. You add nitrogen and phosphorus, there is a shift in genome size towards a larger, more biomass coming off from plants with larger genomes. Of course, we've got a lot of bioclimate data and climate data from all these sites. And so we can start asking questions now about how climate is interacting with this. And what Joe has found is that when the areas with higher rainfall, this effect starts to diminish. So in this model here, you can see that there's interactions between nitrogen and phosphate and precipitation, which reduces this effect size, and also nitrogen and precipitation. So water is ameliorating this effect to some extent. We don't know why. Okay, so it looks like we've got a trend going on here, and it's obviously quite an interesting trend where genome size is affecting the competitivity of these plants. And it goes on really an astonishing scale. This actually quite surprised me, this work. Marie here, another one of our lovely students, has looked at the distribution of angiosperms across the UK as published by BSBI. And they go out and for the last 30 years, they have made every decade a survey of the species of plant that occur in each of the hectads across the British Isles. And you can go to those hectads, every hectad in Britain, and ask what species are there and what are their genome size, and take a mean of it. And so that's what Marie did. And then she plotted it and asked, how did those genome size change over the last 30 years? Have they changed? And on the left here, you can see how it changes between the 1980s to the 90s and the first decade of the 2000s. And you can see that, there's lots of orange colors there and some blue colors as well. As you move to the right here, there's much less blue appearing. It's looking like bigger genomes. Well, they are, as you go through the decades, the bigger genomes, the orange colored genomes, are increasing in abundance or in these hectare squares across the British Isles. And again, you can do bioclimate data and ask what's going on. And the thing that fits the data the best, that explains the data the best, also includes a model on nitrogen deposition. And that as nitrogen deposition has got increased, so has the genome sizes, the species, the bigger genomes, across the entirety of the British Isles. Well, that's looking like a very interesting trend. And Ilya and I then went to Kruger National Park and we thought, aha, they've got a nutrients experiment there. We're going to see what's going on. And here is no nutrients and no water. So on their plot, it has lots of nutrients and lots of water. We're going to get huge, humongous biomass of plants. Oh, nothing at all. So it made me realize I don't understand anything about grassland ecology in Africa. Nothing whatsoever. So there you are, things are complicated. Okay, so there we have we can say about ecological filtering based on genome size and polyploidy. So there are effects. Now I'd like to tell you about the genome downsizing after polyploidy. So we can see that there are effects of having big genomes and that probably they're not going to do so well when the nutrients are absent, and most ecosystems don't have many nutrients. This is a human-made thing to have tons of nutrients kicking around. Most ecosystems are low in nutrients, so that's probably going to disfavor large genome polyploids. So let's look at this distribution of genome size and this skew to the left. This skew to the left is really strange, particularly for angiosperms. And the reason it's strange is because we know that in the divergence of angiosperms, there's loads of polyploidy events. So if we look here, this is a phylogeny of angiosperms turned into a circle. You know, there's the grasses there and so on, all these different groups, all these different major lineages of angiosperms. And all these little green spots all over it, these are hypothesized polyploidy events in the evolution of angiosperms. Now, remember, each polyploidy event should trigger a doubling or more, depending on how many rounds of polyploidy of the genome. So we don't really expect to have lots of tiny genomes. We expect actually to have quite a lot of big genomes. And so that's, it's not sort of apparent what's going on. In fact, if you reconstruct genome size of plants, and this is some results from Jaume Pellicer, what you have here is that the genome sizes are small across angiosperms. So they're small. The colors here tell you their size. So at the left here, the blues and the green colors are small genomes, and the yellow and orange colors are big genomes. And the reconstruction of genome size for all of the monocots is small. Sorry, it's also small for the early diverging eudicots, for the core eudicots, the superrosids, superastrids. Everything has a small genome, despite all those polyploidy events. The ancestral genome size reconstructs at 1.8 gigabase 1c. That's what we think probably the ancestral genome size was, somewhere around there. These polyploidy events are everywhere. Let's just drill into one lineage a bit more. This is the leading to the Poaceae. There's the Rose, Sigma, and Tau events, all polyploidy events leading to the grasses. Any sign of polyploidy in there? None whatsoever. This is our distribution of genome size once again. If we actually pretend that we do some genome doublings to within the monocots from those assumed polyploid events, that's the genome size we would expect for polyploids, for monocots. You can see it's further to the right, it's less skewed, and there are some various bumps in there. If we look at those bumps a bit more, you can see there are bumps here that are not here. And there's this long tail that's not here. And so it's looking like, everything's looking like, following polyploidy, genomes return to a diploid-like form in genome size terms. If we superimpose what we would expect the genome size on a phylogeny here with like this phylogeny here, where each of these little squares represents a hypothesized polyploidy event leading to these species at the top, and we think we have an ancestral genome size of 1.8 gigabases, which we had before. Every time we pass one of these little bubbles here, these little blocks here, we can increase the genome size, assuming there was no genome downsizing. And then we get all these colors appearing. As the genomes get bigger, the color changes. And so by the end here, we'd expect some with very large and extremely large genomes. But in fact, if you look at the top here, the white bar says, no, they're all very small. So there's no sign of these polyploid events manifesting itself in the genome sizes. And so we're back to that picture I showed you at the beginning from Jonathan Wendell about what he calls the wondrous cycles of polyploidy. And this is it. Genomes double to become polyploid. You then get gene losses occurring because there's gene redundancy. And you get these little gaps appearing where genes have been lost. And then you get genome downsizing so that the genome gets smaller. And then you get genome rearrangement, and then this thing looks very much like that thing, and the thing cycle happens all over again. And so angiosperm evolution is having a series of cycles occurring like this. We wanted to look deeply at this side of it, the genome downsizing, because we wondered if things like the nitrogen and phosphorus things were driving that genome downsizing, given that it is also an ecological filter. Maybe that's where the selection pressure, if there is one, is coming from. So we wanted to know how fast that genome downsizing occurs. And what we can do is we can go to phylogenies and put on, we know where the polyploidy event was, take the polyploids out, use the surrounding diploids to estimate the genome size of the ancestor that went polyploid, and work out the rate of DNA loss down this branch. So let's follow it for this juglans species here. So we can reconstruct, well, it's a 50 million year old polyploid, we think. We can reconstruct the genome size before polyploidy to be about 712 megabase pairs. We double that because it's a polyploidy event, so we're now in that grey box there. And we can compare that with the actual genome size, which is 620 megabase pairs, which tells us it's lost 16 megabase pairs per year, per million years, which is 482 base pairs per generation. Not happy with that number, I have to tell you, because that is a small number. We can do this iteratively in the same data set, and you end up with these data. So this is Elaine's work from our group 2. And what Elaine has shown is that the numbers are all small. They're all down in this from this phylogeny, but they're small. Rather too uncomfortably small to be driving selection for genome downsizing. I'll come back to that in a minute. So let's talk about evolutionary selection on genomes. Well, can it be photosynthesis and water use efficiency? I've already told you that the guard cells, for example, can be very different depending on genome size. And of course, that affects pore size. The pore size can affect the amount of water that's lost. It affects the speed the stomata can close, and all that can affect water use efficiency. It also affects CO2 exchange properties within the leaf and between the leaf and the outside world, and that can affect photosynthesis. We've already seen that coming through with the AMAX data and with the water conductance data. And you could start to hypothesize, and here's Lubna's Faizula's work showing how you can hypothesize how water availability can act as a selection pressure on genome size, in theory at least, if only because small stomata can close quicker, so you can react more quickly to environmental changes, you know, a hot, dry day and rain and so on that occurs in a day, and you can open and close your stomata much more dynamically if you have small stomata. And of course, the amount of water that's being lost from the leaf affects transpiration, and it affects photosynthesis, and all sorts of trade-offs going on, which you could easily envisage acts on for selection on genome size. The other thing we can envisage coming from those ecological experiments is that nutrients are affecting genome sizes. We know that nucleic acids are very, very rich in nitrogen and phosphorus, and it could be that that nitrogen and phosphorus is actually a sink for nutrients, of which then have to be satiated before a plant can grow. So maybe just the simple nitrogen phosphorus demands of the nucleus is restricting large genome plants under low fertilizer conditions. And you can easily imagine how low nitrogen phosphate availability then leads to trade-offs for selection for small genomes, because that same nitrogen phosphorus is needed for photosynthesis. And also, just to throw in how complicated things are, of course, water availability also affects nutrients, because if you don't have any water, if you have plenty of water, you can transpire. If you transpire, you can lose, that transpiration process drags nitrogen phosphorus from the roots to the leaves. So you need water lost in order to access the fertilizer. So everything's interconnected. So let's imagine then nitrogen phosphorus is important. You can see here's the genome from Paris japonica. That's the biggest plant genome, and at the same magnification, Genlisea chromosomes, those tiny little things. It's not hard to imagine that this nucleus requires an enormous amount more nitrogen and phosphorus than that nucleus. And on top of that, we know this is a result from Fritillaria imperialis, which has a very big genome. But as the more abundant repeats are, the more they're transcribed. That's what here, this is the transcript proportion of a repeat, and these are the genome proportions of the repeat. When you regress one against the other, the more common a repeat is, the more transcripts there are in the transcriptome. So every time you have a repeat in the genome, you're going to end up with the repeats in the transcriptome, and they're adding more nutrient costs. Okay, so there's a lot of drag going on in nutrients related to genomes. And all of these, individually or collectively, can be selecting for small genome sizes. But is selection possible? Well, perhaps. I mean, here's some very nice result from Kelly et al. And what from Kelly, pardon me, in 2018. And what Kelly showed is by plotting photosynthetic nitrogen use efficiency against selection on nucleotides for low on amino acids for low nitrogen use. What you observe here is if you have a small number here, then it means they're using a lot of nitrogen in photosynthesis, and as you move across this x-axis, they're using fewer nitrogen, so this is more nitrogen use efficient plants and less nitrogen use efficient plants. And if you regress that against selection for amino acids for low nitrogen use in amino acids, you can see that when you have poor nitrogen use efficiency in photosynthesis, you end up with the strongest selection on other on the amino acids in the genes in the rest of the genome. So this is an evidence suggesting that there can be direct selection acting on the genome, but it doesn't based on nitrogen availability and nitrogen use efficiency and so on. But doesn't mean it's happening for the whole genome size. And now I need to go back to the Lubna's data again. And here you can see, to Elaine's data. And here you can see her numbers. These are base pairs per generation lost. They're small, they're in the tens and the hundreds, against a background of 100 million to 10,000 million. It's really impossible to imagine that such tiny numbers can offer a selection pressure to drive genome downsizing. So we've got this ecological stuff that seems to be fitting, but it doesn't seem to be fitting in an evolutionary sense. Doesn't look like that can be visible to selection to me. One way out of that problem is to say, is it punctuated? i.e., the moment you make a polyploid, genome size plummets really quickly. Perhaps that happens. And then after it's plummeted, it just tootles along, drifting at some small genome size. So let's see if there's any evidence for that. Well, we looked through the literature, couldn't really find any for that amongst the very few, relatively few analyses that have been done, but there is one. Here's Tragopogon from the Soltis group by Pires et al. And what they showed is that if you look at populations of Tragopogon miscellus, there is some evidence. These are very young populations of young allopolyploids, and probably 40, been 40 generations for perhaps too many years, but perhaps about 45 generations, some low number of generations anyway. You can see that if you look at those, you know what the parents are because, they're such young polyploids. You can say the parent genomes are like this and the polyploid genome is this size, and they are smaller than you'd expect for Tragopogon miscellus, but not for this one called Tragopogon myrus, which is exactly the same. Elsewhere, you can find examples of upsizing and down. Basically, there's quite a lot of chromosomal change happening in young polyploids, And it's more about sort of sorting out their meiosis and fitness than it is about selection on genome size, I think. So I don't think this punctuated, this fast loss at first, followed by slow loss is going to work. So it remains hard to see how genome sizing with selection acting on it can work. So the other possibility is that there are just other processes happening where genomes are downsizing, not under selection, but for other reasons. And I would like to look at them, whether they're byproducts of other things. So perhaps a byproduct of DNA repair pathways, or simply the abundance of repeats in the genome. So Elaine started to look at that. We haven't quite finished this yet, but this is what she's doing. When DNA gets broken, and it breaks a lot, it needs to be repaired. When it's repaired, there's a whole load of pathways that can repair it. There are homologous recombination pathways, which basically do an excellent job, and DNA is not lost. But there's a whole lot of pathways called non-homologous end-joining pathways, and also some homologous recombination pathways, which are messy, and they do cause DNA loss. So what could be happening is when you go polyploid, the genes involved in these pathways become out of synchrony with each other, or their balance of which pathway is being used shifts. And that polyploid, you may be shifting towards these pathways which that lose DNA. And the thing is that DNA breaks at random and everywhere in the genome. And when you're a polyploid, everything's in duplicate, loads of gene redundancy. And it simply doesn't matter if a gene gets badly repaired and gets disrupted by bad repair pathways. This could whittle away the genome. To a point comes where redundancy is lost, in which case there's selection against that, and then more selections for faithful homologous recombination pathways. So that's the hypothesis she's testing. And my intuition is the reason for downsizing is going to lie there. And also perhaps here, if I go back to these pictures here, this Paris japonica, I mean, I'm just drawing a line here. I'm basically going across tens of thousands, millions of repeat sequences. Those repeat sequences are very readily shed from the genome by recombination processes. There's a lot more you can get rid of here than you can here. So just the sheer scale of having a lot of repeats can trigger downsizing because you've got more targets for dumping repeats. I think, but this is nothing to do with selection. This is underneath by the dynamics of repeat elements. But there are some emergent properties from it. Even if there's no selection action on genome downsizing, it looks like genomes are downsizing. And the effects of that is to reduce their trait space of species with large genomes. So if we look at this here, this is that stomatal density size analysis that we showed you right at the beginning. And when you have a small genome, your stomatal size can be everything from small to very large. That is developmentally regulatable, as is stomatal density. As your genome size goes up, you can't get so small. So your density, size space, trait space diminishes. And whilst development can move you around in this space, it can't move you outside that space. And so one problem with a large genome is that it probably reduces trait space, and that reduces the availability of niches it can live in. And so there are emergent properties of genome downsizing in that it opens up a larger trait space, but it probably was never driven there by selection. So in summary, there does seem to be ecological filtering going on, and that genome size does indeed influence trait space. So it affects niche communities and competitivity. This genome downsizing is widespread across angiosperms, and most angiosperms have small genomes. It looks like this DNA loss is small per generation. It's hard to see how selection can act on that. It's unlikely that it's early in first few generations, but I guess we can't rule it out. It's probably, in my view, a product of other processes that act to reduce the genome size, and that has fitness advantages, but as an emergent property, because it can improve nutrient use efficiency and improve water and transpiration effects. Anyway, that's what we have to say on genome sizes, polyploidy, and plants. Thank you.
Yeah.
Have any questions? Do you want to start the first? Or?
Yeah.
Well, thank you. That was absolutely fantastic. And I certainly have a host of questions.
Yeah.
Oh, yes. So thank you. That was an absolutely fantastic talk, covering a huge range of topics. Does anybody in the audience have a question? Yeah, George.
Thank you, Professor. I was just curious about this. I'm just in the first year of biology course, but for polyploid itself, is there a reason why it is so common in angiosperms? Is this simply a byproduct of how the genome is structured? Is there a selection mechanism that favors it appearing in the first place in this image?
Yeah.
Could you repeat that?
Yeah. So the question, I think, is why is polyploidy so common in angiosperms? Okay, the first answer to that is polyploidy is being experimented with in evolution everywhere. I mean, you can see this, you can see it even in humans, where sometimes triploid babies can make full term, although they die at full term. So evolution is being, polyploidy is being experimented absolutely everywhere. It's also the case, we know that polyploidy is less damaging usually than aneuploidy. It would seem that from the human story, if we had to talk about that again, that, you know, a human triploid, full-term baby, is essentially got one of every chromosome, and yet it's completely lethal aneuploidy, except for chromosome 21, the sex chromosomes, and to a lesser extent, some of the other group D chromosomes. All the other chromosomes completely lethal. And yet, if you amplify them with every other chromosome, it's less damaging. So polyploidy is not as damaging as aneuploidy, is the conclusion you have from that. We also know that with polyploid plants, the higher polyploidy level they are, the more they can be aneuploid. And this is probably because the balance of proteins they're working often as consortia proteins in macromolecular complexes, and they need the right balance of proteins. And if you skew the balance by having an extra chromosome, it messes up the complex. But if everything is amplified, the complex is in an equal stoichiometric balance of one thing to the next, it still works. Okay, so it's happening everywhere. It's not as damaging as you might think, but it's less damaging probably to plants than animals. And when I say animals, I really mean mammals and birds, because there's plenty of polyploidy in fishes, there's plenty of polyploidy in amphibians, you see it in reptiles, and when you move down into the world of worms and such things like that, you'll find polyploidy. Very little work done in some of these invertebrate groups, but I'm pretty safe I will predict they are very common in these invertebrate groups. And the more exquisite developmental processes become, the less common it is going to be found that it survives, because our developmental processes, which make us mammals as complicated as we are really highly tuned gene regulatory systems that get perturbed and we're dead. The angiosperm system is not so exquisitely developmental. It has its moments in flower making, of course, but its real way it survives the world out there is through biochemical complexity. It has to live where it is. It's got to survive drought, it's got to heat, it's got insect damage, herbivory, fungi, everything there. And it does this by throwing biochemistry at the problem. It's not a developmental issue, it's a biochemical problem. And the biochemical problem is facilitated by gene duplication, because when you duplicate genes, you can then diverge another copy of the gene for a new function. And then you can have two ways of tackling a problem, not one. And so that's the lovely thing about gene duplication. Whereas it disrupts developmental pathways, it can enhance biochemical complexity. So I think this is all my conjecture pretty well, but I think that's the root of the issue. And so you see it, I think, where developmental complexity, where there's biochemical complexity is an advantage, and you don't see it so often when it's developmental complexity. But that would be my answer to that.
Excellent. We have a fair few questions online, so maybe
Yeah.
I'll read some of those. So one that just popped up is noting genome size affects stomata-cell operation. Could it also affect sexual fitness and reproduction, such as the ability of pollen grains to be transported between individuals?
So it's a question about weight and transmissibility.
Yeah, I suppose so.
Yeah. It's a good question. I don't think we have any data on that, do we, Ilya? No, I don't think we do. It's a very good question. You can actually make the question a little bit more, we can extend that question. So you could argue then that a larger pollen grain may be heavier and maybe have different dynamics in the air, and perhaps are less likely to be wind pollinated, or at least change its dynamic. It might be more attractive. I mean, a pollen grain, if it's full of big genome, it might be more attractive to insects that are feeding on pollen because they're going to be more nutritious potentially. Yeah.
I just remember the study that within species showing the same sort of pollination biology, you often do get relationship between bigger genomes and bigger pollen, but across the global analysis, because there's really strong selection pressure to keep pollen not too big, the relationship completely falls apart. So there may be lineage-specific cases of that, but not across all angiosperms.
So I think what Ilya's just said is that on a global analysis of genome size against pollen grain size, the relationships fall apart. So I think there are lineage-specific examples you can find, but not on a global analysis. But there still must be a minimum cell size issue going on. But perhaps it's not going to affect the dynamics of the pollen enough.
Is there another question?
But it's a good question.
Is there another question from the audience, or should I continue with the ones online?
Hi, Andrew. I'm wondering about the data. I'm going to switch back to ecology now from development to biochemistry. But I was wondering about the data from Britain showing that the increase in those high genome size plants through time. Is that an effect of one of the big 5 drivers of biodiversity loss, which is nitrogen and phosphorus pollution, or is there something else going on? What do you think is driving that?
Well, we're sort of getting towards the tail end of this. Oh, sorry. So Sandy Knapp just asked the question, what is the driver of the increase in mean genome size in the hectads across Britain over the last 30 years? So to remind you, the hectads of Britain have got the species names put against them through very good botanists who've gone out and recorded what's on there in their area. And we've taken the mean genome size of those areas and plotted them on a UK map, and then asked how has it changed over 30 years. And what you can see is that bigger genomes the mean genome size of the plots across Britain in large part are increasing in 30 years. And so Sandy asked, what is the driver of that? Well, we've got the best driver at the moment seems to be nitrogen deposition. That is driving that. We also know from these data, of course, that and from other data, that global warming is pushing species northwards, and that is quite apparent in these data too, and that the archaephytes, the sort of the originals, are actually diminishing, and in fact, they're moving south. So they're doing strange things, the archaeophytes, but they're diminishing. Whereas our invasives and our neophytes and things like that, they're all marching north. So we've got that pattern happening. And at the same time, we've got nitrogen deposition happening all over Britain, globally, of course. And of all the drivers on genome size, the things that's coming out as the most significant is nitrogen deposition. So yes, us driving cars around, us putting fertilizer on fields is basically nutrifying the entirety of the British Isles. And it's having the effect, astonishingly, at a hectare, which is 10 kilometers by 10 kilometers space, it's having an effect that's pushing the genome sizes up. I was really, as I say, quite surprised that came through as significant, but it is.
There's a question online that I think is related, which is how do you separate out the effects of the nitrogen phosphate from the effects of competition from plants that grow well with that, with the addition of nitrogen?
Yeah, okay. So it is a good question. And in the end, it's complicated on these hectare analysis, because within the hectare analysis, of course, a hectare, 10 kilometers by 10 kilometers, at one side of it, you might be on a bit of chalk ridge with a chalk grassland community, which may descend onto a bit of clay, which is, perhaps an oak forest or something. They're very different communities with very different species compositions in them. Of course, the dynamics within those communities will also affect this pattern. But despite all of that, you do get this significant effects of nitrogen deposition going on. Perhaps then in all these communities, it's rising. Perhaps it's just some of these communities, but we need more better, higher granularity in the data to actually start unpicking community ecology things from those data. However, BSBI also have 1 kilometer by 1 kilometer squared data. It's not as complete as the hectare data. And when we drill into that, which we haven't done yet, we might start to, because at 1 kilometer by 1 kilometer, you can start looking at bogs and, I don't know, etc. It's a small enough scale where you can bring community ecology in as well, but we haven't done it yet.
There's one more question online that I'll add in here, which is, are there estimates about what portion of the nutrients used by a growing cell are used in the nucleus versus the rest of the cell?
Okay, really good question. Yeah, okay, so I did, and I'm pleased that was asked. I meant to say it and forgot. When you have a big genome, that obviously has a lot of nutrients, has a big cell, but it may be at the level of the whole organism, it makes no difference whatsoever because they have less cells. And so end product is you've got a leaf with bigger genomes, bigger cells, less cells. There's no different in nutrient balance to more cells in a small genome. Okay, so you therefore need to extend it to the whole plant, which we haven't done yet, not fully, properly. We're on that case. And of course, we then said, well, surely we know that question. I mean, what is the balance within the cell of all the proteins, lipids, blah, 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 all these, and nitrogen phosphate. Surely we know that. Well, it turns out we have no clue, really. I mean, you can look at it for Drosophila for that thing, and Caenorhabditis for this, and everywhere there's bits of data, right, from different organisms, but there's nowhere you can get it all tidy up together with one thing. So it's really hard to answer that very basic, simple question. Yeah.
Amazing. I have a question. If I don't see any hands pop up, I'll go ahead and ask mine, which is, you talked about, trying to look at kind of the rate of evolution in genome size, and how, your two sources of evidence are essentially, ancestral reconstructions based on extant things, or looking at kind of short-term evolutionary change. But of course, there is a great fossil record of stomata. I know, obviously, I work on animals, so I don't know that much about it, but I know people have talked about stomatal density a lot in terms of climate change. Has there been any attempts to try to look at change in stomatal cell size
Yes.
as it relates to genome size in fossils.
There has been, and there's some very nice work on it by Beerling and co. There isn't enough work done on it. We need a lot more work. We have tried to get that funded, but have been unsuccessful in the past with Richard Bateman at Kew. It is a really good question to go to the fossil record, where we've got ideas on, we've got knowledge of paleoclimates, paleo CO2 levels, and we know enough about the lineages of plants there to have a pretty good idea what they should be, you know, taxonomically. And so, yes, the leaves are perfect tissues. They're good fossil record. Everything's teed up beautifully for that question, but unfortunately, it hasn't been done anywhere. But anywhere in the world could do it, and it's not even an expensive project, because you just need a very good paleobotanist. Well, I mean, that's probably our biggest hurdle. You need a good paleobotanist and a microscope, and to measure stomatal sizes, and then tie that in with phylogenetics, datings, and so on. It cannot do other than be an interesting outcome from that. Yeah.
Any more questions from the floor? Yeah.
The question of stomata brings up a little assistance that I gave years ago at the herbarium at Kew. I used to be there as specialist in conifers. And this was a research that wanted to know whether stomata in conifer needles had increased in density or decreased in density over time, or indeed also stomata size. And we could do that because the herbarium contains specimens of at least 150 years back up to the present. And we chose an area in the world where industrial pollution had been unlikely, and this was the northern part of Canada. Yeah. And so we sampled specimens from the northern part of Canada, and this lady researcher took that with her, the little needle, and the results of it were indeed that over these 150 years, the density of stomata in the needles had decreased. And also there was a tiny change in size, but that was not significant. But the density of the stomata had decreased. And the theory behind it is, of course, that when there is more CO2 in the atmosphere, the needle, the plant needs fewer stomata per area. Wow. So that is a more recent time scale, but I'm sure that can also be demonstrated in the fossil. Yeah, interesting. Do you think people heard that from Maple?
You could maybe give a quick summary.
A summary. Okay, so I just heard from our Swedish colleague. I'm sorry, I didn't quite catch that, but I guess it was a Kew scientist that 150 years of herbarium specimens at Kew were examined to look at stomatal size and density, and there's some indication that density has dropped, and that you could argue that increasing CO2 levels means that a lower stomatal density means they get the carbon the plant needs with a lower density of stomatal pores. Intriguing if that occurs in 150 years. But in fact, I think we sometimes underestimate how fast plants can respond to things. That could be a developmental thing rather than a hardwired thing. Yeah.
Making stomata is an expensive.
And of course, developmental things can be switched, as and when they're needed. So yeah, it's very interesting.
There was maybe one last question, which was posted online, which is whether genome size correlates with anything else other than stomatal size and density, any other organelles or anything like that.
Yeah, well, I said in the talk, it links with vein density, and so that has issues related to water conductance. Do you know, I'm going to hand over to Ilya. She's the expert on what it correlates with.
Well, I think at the nuclear level, this correlates with scientific work because we were
You can repeat it. Special guest appearance. Oh, that. You have to go stand in the laptop.
Just to say that at the nuclear level, we see effects of genome size correlating with both the size of the nucleus, the volume of the nucleus, as well as the timing, the duration of mitosis and meiosis. So there are effects at the nuclear level. Andrew's already spoken about the effects at the cell level, for example, in terms of cell size, stomata. Stomata are one of the cases where it correlates very nicely because stomata don't seem to produce vacuoles often. So as Andrew said, if you have a small genome, you can make bigger cells by vacuoles. And so the correlation often breaks down in cells which are specialized for particular functions. But for stomata, which of course are specialized cells, they don't seem to produce it. So you get a good correlation there. And you also get a good correlation in young rapidly in dividing cells because they also are not producing vacuoles. Then you have these effects, as Andrew also mentioned, a correlation with different tissue types. Because of the packing density, you can't pack lots of big cells together, so that affects tissue density like vein density. There's the physiological effects, and then there's lots of correlations at the whole plant level of genome size being correlated with where plants are able to grow and how they grow, the type of life cycle they adopt. Species, for example, with small genomes are effectively restricted to having small genomes, but that's sort of as a consequence of the knock-on effects that you see at the cell and the nuclear level, because if you want to be an effective invasive species, you want to be able to grow quickly, produce lots of seeds which are small that can disperse all over the place. All of those are correlated with having a small genome size and not a compatible with a bigger genome size. So you tend to see that invasive species are characterized by small genomes, and it goes on and on. There are lots of different correlations, but they all sort of have these, they're grounded in these effects at the nuclear and cell level. But they often are lineage specific. They're not necessarily, because the particular genome size of a species is really as a result of many different trade-offs and selection pressures or not selection pressures, depending on what has caused it to be at that level. So what you see in one lineage may not necessarily apply to other lineages, but there is sort of overarching things.
Excellent.
Okay.
Okay. Well, thank you very much. Okay, any final questions? Or actually, we're a bit over time, so I think
Okay.
we'll thank Andrew again
Thank you.
for an excellent talk. And I think that kind of final interaction there was one of the really nice things of the Linnean Society. I know many of the people here in this room are already part of our fellowship or members of the Linnean Society. We have a huge online audience today as well. So for those of you who don't know about the Linnean Society, that interaction is very much the sort of thing that happens here. This is a society for people who are passionate about the natural world, who believe it should be understood, valued, and protected. And regardless of whether you're a scientist or an artist or historian, or just somebody who really enjoys nature and wants to work towards its preservation and improved understanding, we would welcome you to join the Linnean Society as a member or a fellow. There are links posted in the chat if you'd like to find out more, or if any of you are here, just grab us during the reception. You can sign up today if you like. And if you want to hear more about the events coming up at the Linnean Society, the next few events, it's actually a very action-packed December coming up, which is just a few days away, of course, December. So our Founders Day lecture is coming up next Friday, and this is on Linnaeus and human diversity. So really tackling some of the, I think, tough questions related to, obviously, Linnaeus and his history. And that is on Friday, December 2nd by our own Isabelle Charpentier, as well as Stefan Mueller from Cambridge. Then on December 8th, Thursday, December 8th, we have the Irene Manton lecture, which is held at the University of Manchester, but also online. And this time we have Rebecca Willis, who's a professor in energy and climate governance at Lancaster University, talking about, can democracy save us from climate change, which is a really fascinating question to ask. And then finally, on December 15th, is it? We have our Christmas lecture called A World of Urban Birds by David Lindo, who is last year's H.H. Bloomer Award winner. So please join us for any of those events and look forward to speaking to some of you upstairs in a few minutes. Oh, sorry. And then for those of you who are here today, please join us upstairs for the reception. And if you are interested, there is some Linnean Society merchandise available for sale. Okay.
Thank you.
Thank you very much.
Thank you.